after that.
Okay, cool.
Okay, maybe get started. Good morning, everyone. Today we have Professor Ludovic Vallier from University of Cambridge. He received his PhD from École Normale Supérieure of Lyon, University Claude Bernard in 2001, where he studied the function of cell cycle regulators in pluripotency. He then joined a group of Professor Roger Peterson at the University of Cambridge, Department of Surgery, where he uncovered the mechanisms by which TGF-beta signaling controls self-renewal and differentiation of human pluripotent stem cells. He became an independent investigator in 2008 after receiving a MRC non-clinical senior fellowship and was named Professor of Regenerative Medicine in 2015. He recently joined the Berlin Institute of Health as Einstein Professor for Stem Cells in Regenerative Biology. His group, based at BWH Center for Regenerative Therapy, takes advantage of human pluripotent stem cells and primary organoids to generate liver cells with a clinical interest for disease modeling and cell-based therapy. The floor is yours, Professor.
Thank you, Professor Domke. It's a great pleasure to be with you tonight, for me tonight and for you today. And so today I'm going to present the work that my lab have been doing the past few years on the organoid and pluripotent stem cells to study liver biology and disease. And so, as it was said in the introduction, I recently moved from Cambridge to the Berlin Institute of Health, where I now have an assistant professorship, and I have also an affiliation with the Max Planck Institute for Molecular Genetics. Just before to start my talk, I just would like to say that I'm recruiting postdoc in Berlin. So, if you are interested, please drop me an email or apply to the postdoc link that are here. If you Google me or Twitter me, you will find my new address and this information. Also, before I start, I need to just mention some conflict of interest. I've been involved over the years in the creation of several companies called Definition, Aculive, and Biliary. And part of the data I'm going to present today have been generated either with the support of those companies or in close collaboration. Okay, so that's just to give you an overview of the goal of my lab. Really, the goal of my lab is to use human pluripotent stem cells and primary organoid. And when I say primary organoid, it's organoid derived directly from primary tissue. Is to use those two model systems to produce liver cells in vitro and to use those liver cells for this modeling, cell-based therapy, and also to study mechanisms that control endogenous regeneration of the liver. And so today I'm really going to touch base on this modeling and cell-based therapy, and I will not talk too much about the work we're doing currently on endogenous regeneration. So I'm going to start by introducing the liver. So the liver is the biggest organ in the adult body. It fulfills key vital functions, including detoxification, drugs, secretion of key protein, albumin, the most abundant protein in the plasma, for example, also factors. Key metabolic organ is processed lipids, store glycogen. And so this organ is really a filter of the blood directly and make sure that the blood now is detoxified from all toxin and bad component that you can have. And the main functional unit of the liver is the hepatocyte. So that's the main cell type of the liver. It's represents 80 to 90% of the liver mass. And these cells fulfill most of the function I just mentioned. So really is the key unit of the liver. The other cell type, which is really important, is cholangiocyte. And the cholangiocyte, in fact, have cells that line up the biliary tree. The biliary tree is a network here in green that you can see that basically drain the liver from all the toxins that are produced by hepatocyte and especially bile acid. And the biliary tree is connected to the gut, and basically that's allow, in fact, everything that is produced by the liver as bile to be excreted in the gut. And so those two cell types are really the key unit on the liver. 100% of the disease that target the liver will target either hepatocyte or cholangiocyte. So the key interest in the liver, the drive beyond looking at liver, is that liver disease is one of the rare group of disease where mortality is still increasing year on year. And that's simply explained by the fact that during a long time, liver disease have been a bit neglected because of the main drive being alcoholism, so a self-infected disease. That started to change with a disease that I will mention several times in my talk, with the apparition of non-alcoholic fatty liver disease, which a disease that is not associated with alcoholism. And because of this lack of funding and general interest in liver disease, that's mean that therapy for liver disease have been a bit delayed. It's changing very quickly and very rapidly, but nonetheless, we can see the impact on the mortality of the disease. And because of this delay also, there is currently no treatment for end-stage disorder, for end-stage liver disease. The only treatment is liver transplantation, which of course comes with limitation, including the use of immunosuppression, which have important side effects and can induce infection and cancer. But also what is the biggest challenge with liver disease is that the two cell types that I've mentioned, hepatocyte and cholangiocyte, are fairly complicated to grow in vitro. Okay, so if you take hepatocyte out of the liver, after a few hours, they will start to differentiate and lose their metabolic function. So that's means that currently we can't grow human hepatocytes in a dish, which of course limits the number of screen you can do, for example, for toxicology screen, but also disease modeling and also cell-based therapy. And cholangiocyte is a bit similar, but I will come back on that and I will show you that now we can grow cholangiocyte cells in vitro as organoids. So to address this limitation or this difficulty to grow liver cells in vitro, what we decided to use is human pluripotent stem cells that are either derived from human blastocysts, so in this case, human embryonic stem cells. And what we used to do in the past was to isolate the inner cell mass of a human blastocyst and grow them in culture. And sometimes, it was a very inefficient process. We were able to derive a cell line that we call human embryonic stem cells. Of course, these human ER cells are now being slowly but surely replaced by human-induced pluripotent stem cells that generated by direct reprogramming of somatic cells using the famous 4 transcription factors discovered by Yamanaka and from which he got the Nobel Prize. In this case, those cells generated by direct reprogramming, it's a very efficient process. Now we can generate iPS from almost any patient and from a broad number of cell type. And those cells also biologically can be grown in exactly the same culture condition that we grow human ER cells. In fact, in our hands, we can't distinguish human ER cells and human iPS. So that's why now we biologically mainly focus on human iPS in the lab. And the new system that we are using in the lab, or at least new for us, is organoid. And when I say organoid, it's really important. It's not human iPS differentiated in 3D. It's organoid derived directly from primary cells and from primary tissue. It's basically epithelium that we go in 3D using the culture conditions that were initially developed by Hans Kleffer for intestinal organoids. And so that's really the two model system I'm going to use today, and I'm going to explain what we're doing with those cells. So, of course, the first step is to be able to generate liver cells using human iPS. So the big advantage of human iPS is that they can grow almost indefinitely in vitro, and then you can differentiate almost into all the cell types, including hepatocyte and cholangiocyte. And that's been key objective in my lab is basically to develop protocol that allows to differentiate human iPS into hepatocyte. And over the years, we have developed a protocol, which are around 35 days of differentiation in vitro, and which basically follow in vitro the natural path of development that happen during different hepatocytes in vivo. And we differentiate first human ER cells into endoderm, and then endoderm specification forget into hepatic endoderm, and then hepatic endoderm into hepatoblast, and finally into hepatocyte. And then we have a phase of maturation where we see an increase of functional activity in those cells. The cells generated at the end of this process now express homogeneously albumin, key marker, alpha-1 antitrypsin or HNF4, key transcription factor in the liver. This population is homogeneous. Almost 100% of cells will express those markers, and those cells will display some level of activity, including urea cycle, albumin secretion, alpha-1 antitrypsin secretion, also CYP2A4 activity. CYP2A4 being the cytochrome P450 enzyme that detoxify 30 to 40% of the drug that are currently used for treatment. So key enzyme for toxicology. So that's really good. We can produce really large quantity of those cells. And we can also transplant those cells in animal model for acute liver failure. So in this case, we use a UPS kid mice that overexpress a transgene that kill its hepatocyte. And then when that's allow to make space in the liver, we can transplant our cells generated in vitro. And what we see nicely is the apparition of a cluster of human hepatocyte in the mouse liver. And we can detect the presence of those human hepatocytes either by immunostaining or by secretion of albumin. So that's great, okay. But what is also important is that we've done now a large amount of really detailed analysis of those cells, and we know that those cells are not completely identical to adult hepatocyte. In fact, they lack key function. They have a low, they lack the whole path cytochrome P450. They express fetal markers like alpha-fetoprotein, and in fact, they look a bit closer to fetal cells than fully adult hepatocyte. Okay, and that's a limitation for a number of applications, including, for example, for toxicology screen, because we know that our cells are not a very good model for this kind of application. And nonetheless, I will show you that we can use those cells for a number of applications that are still relevant for clinical development. So over the years, of course, we tried a lot of different approaches to improve the functionality of the cells. We tried to screen small molecules. We tried the epigenetic modifiers. We tried, of course, growth factors. We tried different type of extracellular matrix and co-culture. And I'm going to present the work that we've done on co-culture and extracellular matrix to show you that indeed, we can complexify the culture system that we have developed, and that will allow us to improve the function of these cells. Not a magic solution, but nonetheless, it's help. So the system we use for 3D culture of those hepatocytes generated in vitro is the RAF system, which is a commercial system developed by Lonza. And it's we choose this system because first it's based on collagen, which is a known extracellular matrix that support hepatocyte, but also because it's very easy to use and it's commercially available. And this RAF system directly comes in 48 or 96 well plate. And you have a solution of collagen where you mix your cells and your medium in it, and then you come with a buffer and you aspirate the medium so that your cells end up embedded in a semi-solid disc. And that's how we embed our hepatocyte generated in vitro, and now that we can have those cells in 3D. And when we do that, basically, what we see is an increase of function, or we have increase of albumin secretion, for example, and a decrease of fetal marker like alpha-fetoprotein. And when we look at the transcriptome of the cells we growing in 3D, we see that the cells we have grown in the raft are a step closer to adult hepatocyte. So they express better level of detoxification enzyme, drug transporter, and so on. They're still not fully identical to a primary hepatocyte, but they are very similar. They start to be a step closer. And so what also is important that what we see appearing is that those cells in 3D can be grown for a much longer period of time. When we grow the cells in 2D, after 45 days, they will start to die. Whereas with the cells in 3D, we can keep them for 6 months, even 9. They will still display key functional activity like C3A4. And also what is interesting with the 2D, for example, here shown in immunostaining is the expression of this protein here, MRP2. MRP2 is a key transporter, and the cells it's really important because that's this transporter that's going to allow the accumulation of a number of toxins, which induce toxicity in the liver. If you don't have expression of this protein, at least in a polarized way, you can't have accumulation of toxin, and of course you can't model the toxicity. And that's shown here in 2D. We never see this protein properly organized. Okay, another key interesting property with this 3D culture system is that we can model lipid accumulation. And lipid accumulation is a key process in the NAFLD disease, in fatty acid and chronic liver disease. And what you can see here is that cells going in 2D, where we have cells going in fatty acid, which are known to induce lipid accumulation in hepatocyte. And you can see that in 2D we have a bit of accumulation after 1 week. It's very weak. When we go to cells in 3D, we see those beautiful lipid droplets appearing in the cells, showing that really we start to have lipid accumulation. And what was also interesting is that we can also model lipotoxicity, which is the second hit of the disease with palmic acid in this case. And what we see is that we see a strong increase in cell death with this the use of palmic acid, but only in 3D. So that shows that really with this 3D system, we improve the function of the cells, but we can also model some property of the cell that are impossible to model in 2D, including, for example, lipid accumulation and lipid toxicity. So, of course, now the advantage of a 3D system is that also we can do co-culture and mix cells together. And that's exactly what we did now with this RAF system. We started to add the other cell type in the liver, including cholangiocyte, but also cells that are known to be involved in liver disease, like stellate cells, myofibroblasts in the liver, and macrophages that are involved in the pro-inflammation response of the liver. What you can see here is basically the cells are co-cultured together and treated in the raft. In green here, you have hepatocyte cells generated from iPS that express the GFP. In red, you have cholangiocytes that express the RFP. And then you have stellate cells, which express, in this case, that express the blue FP. And unfortunately, we couldn't stand for macrophages in this case. But what you can see is that what is interesting is that the hepatocytes are going to self-organize around the bile duct formed by the cholangiocyte, and that the LX2 cells are going to grow in between those cells. And in fact, that's closely resemble, what the architecture of the liver, because in the liver, cholangiocyte form tube that are surrounded by hepatocyte with stellate cells that are between those different structures. And again, we can co-culture cells for 1 week, 4 weeks, and then for longer. What we see is that this co-culture improves slightly, but still improve a key function of the hepatocyte, including albumin secretion, but also some citric activity. And for the first time, of course, also we can monitor the secretion of bile acid because of the presence of cholangiocyte that capture those bile acids. So again, in this case, we can also model oleic acid and pamine, which are two fatty acids that are involved in liver disease, non-alcoholic fatty liver disease. And what you can see in this case is that those cells, only hepatocyte will accumulate lipids, which we'll see an induction of CYP2A4 because it's known that the oleic acid will induce CYP2A4. And what we also see is a strong cell death used by those treatments seen here. And also what is interesting is that for the first time, we can see a pro-inflammatory response, which is part of the disease progression mechanism, and in this case, it's TNF-alpha. Okay. So what I'm showing you here is with this 3D model system, we slightly improve the function of the cells, but we can also model different aspects of disease, which are linked with the different function of those cells, including pro-inflammatory response, but also for example, activation of stellate cells. So what that summarizes this first part of my talk is that really those 3D systems are not a magic bullet to really improve, generate fully functional cells, but they are a good system to really allow to have a more physiological environment that allow to model different aspects of disease, and especially in liver disease will be lipid accumulation, lipid toxicity, and pro-inflammatory response. So then now, of course, the key question for us is how we can apply this kind of relatively complex model system to inform biology and to understand better disease. And what we're currently doing is to look at, in fact, genetic modifiers that are involved in this progression in human. So non-alcoholic fatty liver disease, as I told you, is one of the main drivers of this increase of mortality in liver disease. And it's induced by fatty liver. It's linked with obesity and diabetes, but there is also an increased number of lean patients that suffer from fatty acid accumulation in the liver. And it's estimated that in the US and the UK, around 30% of the population will have a fatty liver. Of course, not all those patients will progress into a chronic liver disease and a stage disease. But nonetheless, this fraction of patients that progress is increasing. And we know, due to the GWAS studies, that this progression is strongly associated with a key genetic variant. And one of the most common variants associated with progression of NAFLD into a cirrhotic disease is PNLP3. And here I just put a graph here showing you the presence of specific genetic variant in PNLP3 in the population of patients with progressive NAFLD in Allen Brooks Hospital in Cambridge. And you can see that more than 50% of those patients will have this genetic variant. Whereas it's much less frequent in the general population. And that's not for Caucasian population, but we know that, for example, for Hispanic population, this number will even further increase. So that just tells you that really this variant is a key driver of the disease. And surprisingly enough, the function of this variant, the function of these genes is relatively unknown and not fully understood. So what is known is that PNLP3 is involved as a triglyceride lipase, which is involved in the formation of lipid droplets inside the hepatocyte. Okay, so that's well known. The problem is that functional studies on PNLP3 have been challenging. First, because we can't grow hepatocytes in vitro, and PNLP3 is specifically expressed in human in hepatocyte. Second, that mouse models are not extremely good to study PNLP3 because PNLP3 in mouse is not expressed in hepatocyte, it's expressed in adipocytes, so at least mainly in adipocyte. So all the functional studies targeting PNLP3 in the mouse have not generated a good model of disease progression for NAFLD NASH. And so that's why now we decided to look at this gene in the context of human IPS. First thing that we did is to basically generate human IPS line that carry the genetic variants. We use CRISPR-Cas9. We did a point mutation in the PNLP3 gene so that we can have isogenic like wild type and line that carry these genetic variants. And what we also did is a total knockout of the gene so that we could look at the function of the gene and compare this function of the gene to the function of the genetic variants. And that changes here. Here you have basically a qPCR on different exons of PNLP3 showing you the expression of PNLP3 in the wild type, in the variant, and the knockout. Showing you that we do have a loss of expression with the knockout. Then after what we did is take those different genotype and induce differentiation. So wild type, variant, and knockout. Just to check that if those, the genetic modification could affect the capacity of differentiation of the cells. And what we observe is that the genetic variant or the knockout of PNLP3 don't affect the capacity of the cells to differentiate into a hepatocyte. It was great, meaning that we can look now at hepatocyte. And what we did, of course, is to grow those cells in 3D and then treat those cells with oleic acid and palmic acid. Oleic acid inducing lipid accumulation, palmic acid inducing lipotoxicity. And what we observe first is that the cell line carrying the variant or the knockout start to accumulate lipid before even we treat the cells with those fatty acids. And when we treat the cells with fatty acid, there is a strong accumulation here shown by body staining or by FACS, and that's shown that basically the cells either knockout or carrying the mutant display an aggravated accumulation of lipids. But what was even more surprising is that when we treat with palmic acid, so something that kills hepatocytes usually, in fact, we didn't induce cell death, and that's shown here, for example, where you show that the palmic acid is not inducing cell death when you compare it to the control. And what that means and what we observe instead is lipid accumulation. So it seems that palmic acid doesn't induce lipotoxicity, and in reverse, it's induced lipid accumulation. So it seems that in fact PNLP3 loss of function or the genetic variant is protective against the lipotoxicity, which of course go against the notion that PNLP3 variant is important for the progression, as in fact the aggravating factors of the disease. So we did a bunch of wide analysis. In this case, it was lipidomics analysis, which we can do with the cells because we have enough cells. And what we observe with the genetic variant or the knockout is a switch in the lipid species that are secreted by hepatocyte. We observe actually a strong increase in triglyceride with the variant or the knockout, suggesting that the lipid metabolism is strongly affected by this genetic variant in hepatocyte. And what we then hypothesize is that in fact the variant or the knockout could be protective against the lipotoxicity of palmic acid because it's basically transform palmic acid into a triglyceride, which absolutely not the case in the wild type cells. And to demonstrate that, we use a small molecule inhibitor of the triglyceride pathway to show that when we add palmic acid and those inhibitors, we reestablish the cell death that we usually observe in wild type cells. So again, suggesting that this process through this pathway is probably what rescues the cells. And of course, we were banging our hand on the wall because NLP3 is supposed to be a factor accelerating the progression and really important progression. And all of data were suggesting that, in fact, it was protective against the disease. And that's why what is the experiment we decided to do is to basically see if other toxins that are known to damage the liver, like ethanol or like methotrexate, which is an agent used for chemotherapy, which is known to be hepatotoxic. We decided to try now if those agents could have a cumulative effect on the cells with lipids. And what we observed that indeed, no, in fact, those agents are much more toxic on the hepatocyte carrying the genetic variants or the knockout than they are when they are on the wild type. So that means that PNLP3, protects probably against lipotoxicity, but in fact increase the impact, the negative impact of other toxins. And so that's the model we have now. In fact, the patient that have a PNLP3 mutation progress not because PNLP3 affect their capacity to store lipid or process lipids, but in fact they are much more sensitive to other toxins like alcohol or medication or drugs. And that's why often those patients are resistant to any treatment or conventional treatment or dietary change and so on, simply because those patients, in fact, need to be protected against other toxic and not against their fat or their fat accumulation. And what we hope now, of course, it's really important to understand how you can develop drugs against the disease, because that means that you need to manipulate PNLP3, but in a certain way. So, trying to knock down PNLP3 may not be the best idea, because those patients will develop even faster the disease. And also, that tells us that we need to manage those patients a bit differently, and that we should be, focusing on other toxins on those patients rather than focusing only on the food intake. Okay, so I think what I wanted to show you here is just an example of what we can do with the cells now, what are the culture system we can have, and what new type of biology we can develop. Now I'm going to completely, and going to start up with cholangiocytes, the other cell type of the liver. So far I've talked about hepatocytes, and hepatocytes, as I told you, at the moment we can't grow them in vitro. So even if we try to do organoid with hepatocyte, that doesn't work. Whereas cholangiocyte we can. Okay, so my lab and other lab have developed culture conditions that allow us now to take a biopsy from the liver and grow cholangiocyte organoid. So that's exactly what we did. So basically what we have been doing the past few years is to take a cholangiocyte from different regions of the biliary tree, the biliary tree inside the liver, the biliary tree outside the liver, and then derive cholangiocyte organoid from those different regions. And why we did that is because it's known that disease affecting the liver affect the biliary tree regionally, meaning that they will affect the biliary tree that is inside the liver differently than they affect the biliary tree that is outside the liver. And that's something that's difficult to understand, why cholangiocytes that are outside the liver may be more resistant to disease than cholangiocytes that are inside the liver. So we did derive from the different region. It's a very simple process. You can either get biopsy, you can get ERCP, which is a basic examination that the clinician will do. You can also collect gallbladder, which is something that's a resection of gallbladder happen very often because it's not really incredibly useful organ. And from that, then you can basically scrap cells, put them in 3D in Matrigel, and after a few days, you will start to have those beautiful spheroid appearing that we call a cholangiocyte organoid, extrahepatic cholangiocyte organoid or intrahepatic cholangiocyte organoid. And you can derive those cells from almost any patient. There is no impact of age or gender on the capacity of deriving those cells. Those cells can be expanded in vitro for 20-30 passage. Their karyotype is stable, and they maintain an homogeneous expression of biliary cholangiocyte marker. They also express a lot of markers and that are specific for cholangiocyte, of course. That's showing you a comparison of freshly primarily isolated cholangiocyte versus cholangiocyte organoid in vitro. As you can see, there is differences, but these differences mainly cell cycle marker, because of course those cells proliferate intensely in vitro, whereas in vivo they are bioclickers. And what is also important is that the cells will express protein, and they will express this protein in a polarized manner, which is again really important for the function of cholangiocyte. They have also key functional activity. They can transport bile acid. In this case, we use FITC bile acid, which is fluorescence, showing that the cells can exclude bile acid. They can respond to hormone stimuli, so they can swell directly. And they secrete gamma GT, which is one of the main hormones secreted by the cells in vivo. And so now the question that we did ask is, all those cells derived from different regions that we know are affected differently by disease, are they really different? And what is their intrinsic differences that could explain their resistance to disease? So what we said do is to directly derive cholangiocytes from the gallbladder, the intrahepatic region, and the common bile duct, which is directly the tube that links the liver to the gut. And so we did that now. For what we did is to take sample from cadaveric donors, so patients that donate their organ for transplantation, and take now a bit of common bile duct, gallbladder, and a biopsy. And then we derive organoid from the cells, and then we perform single-cell transcriptomic to understand really, if those cells first are different in vivo, if we can derive organoid, and then if the organoid derived from this different region of the liver are similar. And what we observe first is that, yes, those cells in vivo have different transcriptome. They show a regional identity. So the cells that are from the intrahepatic duct or the common bile duct to the gallbladder will display different gene expression. Okay, and like that, we have been able to identify specific markers for each region, like SOX17, for example, for the common bile duct and the gallbladder, or SOX4 for the intrahepatic duct. So that's the first things, really those cells in vivo expression pattern. Then we did the same thing with organoid. And what was surprising with organoid is that, in fact, the transcriptome of those cells is almost identical when we grow them in vitro. So if you generate from the gallbladder, from the intrahepatic or extrahepatic, they will all directly look the same when you grow them in vitro. And that's not was a surprise, because that means that organoid, when you grow them in vitro, contrary to what is the dogma in the field, they lose their original identity. They're not absolutely identical to what happened in vivo. But in fact, no, that's normal, because when you have cells in an organ, of course, the microenvironment will have a strong impact on the transcriptome of those cells. So what we did is hypothesize that, in fact, no, as we take cholangiocyte out of the liver, they lose this microenvironment information, and they basically become all the same, because they all share the same external information with the culture media that we use to grow those cells. And to demonstrate this, what we did is to grow cholangiocyte organoid, and especially gallbladder cholangiocyte organoid, in presence of bile acid, because bile acid, we know, is the toxin secreted by the hepatocyte as a waste product, is very strong, high concentration in the gallbladder. So what we decided to do is to put cholangiocyte organoid in presence of high dose of bile acid to see if this stimuli could directly reprogram the cells, or at least induce the transcriptome of the cells to become more similar to primary gallbladder cells. And that's exactly what we observe. So what you see is the organoid, but then when you treat them with biliary, with bile acid, they become now much more similar in terms of transcriptome and identity to a gallbladder primary cells. Okay, so that shows you that basically when you grow cells in vitro, of course, this cell is going to lose information, going to change this transcriptome, but that doesn't mean that the cells is different from what is in vivo. Just mean that the microenvironment have changed and the cells is reacting to that. And if you modify this microenvironment, the cells will become more similar to in vivo cells. And to really demonstrate that, what we decided to do is to basically show that we could retransplant cells grown in vitro into a mouse model for biliary tree injury. So very simple model. We take a mouse, we inject a toxin in the biliary tree, it kills the endogenous hepatocyte, and then we take a cholangiocyte organoid grown in vitro, human cholangiocyte grown in vitro, and we inject that in the mouse liver. And what we observe is really simple. First, those cholangiocytes rescue the mice, because if you destroy the biliary tree of a mice, it dies in a few days. When we inject human cells, the mice are rescued. And in fact, we can see that here by MRCP, you can see that in fact you have a reconstruction of the biliary system. And you can see human cells engrafted in the mouse liver and basically forming tube, recolonizing the bile duct system. What was also very interesting that we took not only cholangiocyte from inside the liver, human liver, but also cholangiocyte organoid derived from extrahepatic biliary tree, injected in mice. And in fact, we found that those cholangiocyte organoid can basically recolonize the mouse liver as efficiently as intrahepatic cholangiocyte organoid and express the right markers of intrahepatic cholangiocyte. Meaning that, in fact, that show that cholangiocytes inside and outside the liver are extremely similar, not identical, and have the same capacity and the same property in terms of function. And finally, as a proof of principle for future cell-based therapy in the context of liver disease, is to also show that we can do the same experiment in a human liver. So there is now a technology that's more and more used in transplantation, which what we call nomothermic perfusion of organs. So you take an organ, a kidney, but also a liver. In this case, that was the liver. You perfuse it with warm blood. And that's how you basically to first clean the organ from immune cells, but also allow the organ to recover its anti-ischemic. And that's improved the outcome of transplantation. And Cambridge have been leading this time of application, especially for the kidney and for the liver. So what we did is take one of those liver that was not, of course, transplanted, but one of those human liver, we perfuse it with warm blood, and then we basically doing a relatively complex procedure. We injected cholangiocyte organoid grown in vitro in a small bile duct, one of those liver lobes. And what we observe is that indeed, human cholangiocyte grown in vitro, in this case, they are marked with a RFP, can engraft in the human liver and can basically reconstruct a bile duct. And again, that was obtained with cholangiocyte from intrahepatic duct or from extrahepatic gallbladder. So meaning that, again, cholangiocytes are interchangeable and they can be used to replace each other in vivo in the course of cell-based therapy. So for us, this approach now represents a new technology that we can use as a proof of principle to demonstrate that cells can engraft in a human organ, and we hope that will accelerate the clinical application of our cell-based therapy program. Okay, so what I've tried to show you is that basically cholangiocytes are interchangeable. They are the same in the biliary tree. Of course, they will have originality in vivo, but that's imposed by their microenvironment. When you take them out of this microenvironment, in vitro, they will basically lose this regional identity, but they can regain it if you change the culture conditions or if you retransplant them in vivo. Okay, so to conclude my talk, what I would like to do is just to summarize what I've explained. So, we can now generate a large number of cell types from human pluripotent stem cells. But in most of the cell types that are generated from human pluripotent stem cells, there's an issue with the functional maturity of the cells. And that's not entirely surprising, because when you think of what we're trying to do, we're trying to mimic in vitro normal development. And production of a functional hepatocyte, for example, in vivo in human, take 20 months, because in fact the liver in human is fully functional only 9 months to a year after birth. So trying to compress this development in few weeks is of course a massive challenge. So we can expect that we're going to miss a step and that the cells we're going to generate are not going to be fully functional. We can correct that partially pre culture conditions and co-culture. It's not a magic bullet. It's not going to make your cells fully functional, fully adult, but nonetheless, that allows you to really increase the complexity of your cells and allow you to model complex disorder like non-alcoholic fatty liver disease. Of course, now we have other model systems that are starting to take more prominence, and organoid was one of them. So far, we can only use them for cholangiocyte, but soon, in my opinion, we will be able also to use them with hepatocyte. But I don't oppose the two systems, organoid and human pluripotent stem cell. I think they are really complementary. Organoid are more representative of the ideal organ, but they also have the defect that they will reflect the disease, and so they will be damaged in case of disease. Whereas the iPS are more representative of development and allow you to probably look at this progression a bit more precisely than organoid. What I'm trying also to show you is that those cells, and I've only talked about cholangiocyte, but we're doing a lot of work with hepatocyte, and a great opportunity for regenerative medicine. We can now use those cells for cell-based therapy, and we are currently now working and developing those cells at the clinical level, so like that we can use them, into patient. But of course, ultimately, we want also to use those cells to understand more the basic biology of organs, and how disease affect the biology of this organ. And for example, the work I've shown you on Biliary is a good example. By looking at the function of the gene in the context of NAFLD NASH, we also understand a bit better the role of this gene in lipid processing and lipid metabolism. And that's really the combination of all those models, and of course, using again mouse model that will allow us ultimately, to develop new therapy and understand better the liver biology. Okay, so I'm not going to do much of that. I'm just going to thank all the people that have done this work. So that's my group in Cambridge, and the work that I've shown you, most of the work done on P2 was done by Samantha Tilson, which was an NX Oxbridge student, which back at the NIH now. The work on the co-culture and 3D has been done by Carol Amoral, a postdoc in my lab. The work on cholangiocyte organoid have been mainly done by Fotis Samposiotis, who is now leading his own group. And I would like also to thank the founders that support group, especially the DRC, which have been the main funding agency supporting the work of my group for the past 10 years. Thank you for your attention, and we'll be happy to answer any question. Thank you very much.
Thank you for the excellent talk, Professor Vallier. One simple question came in: can we use AI or deep learning to find the NAFLD? Are you thinking of using some of these latest computational tools?
So yeah, that's something that other people are certainly trying. That's we more in the functional validation side than the identification of target or large scale data analysis on patient cohort.
Thank you. One of the main problems at some stage features of liver is having senescent cells. These cells have local effects, and as the individual gets older, the number of these cells will increase. The question is, can we see these cells in human-induced pluripotent cells, or are they homogeneously growing?
Yeah, that's a good question. So, in fact, it's known that in chronic liver disease, and again, it's not a black and white picture, but in chronic liver disease in human, you have senescence is taking place, and the cells stop to grow, especially hepatocyte. So first proliferation of hepatocyte, even in a non-disease organ, is very low, but it's even lower in chronic liver disease. And that's why a lot of people think that regeneration is blocked, because of course the liver is a very regenerative organ. If you cut half of your liver, it will regrow. But in chronic liver disease, this process seems to be impaired, and that's by controlling proliferation of hepatocyte. It's blocked. So the advantage of IPS, derived hepatocyte, that they are quiescent already. There's no proliferation after 25 days of differentiation. So it's a drawback and advantages in some way, but this aspect is already included in our model.
Thank you. When you create hepatic organoids, do you do functionality testing? Like, do you take alcohol and make sure the cells kind of metabolize it and things like that?
Yeah, so we done that for lipids, lipid accumulation, lipid toxicity. Of course, that's a demonstration that the cells are functional, and we have also shown that those cells can, those different cells that's in the co-culture will react to the process. We'll shown that cells can be activated, that macrophages induce having pro-inflammatory response. We know also the 3D culture is very good for cytotoxic studies, because as I show you, the MRP2 protein allow the secretion of accumulation of toxin within the cells. So when you are in 2D, you don't have those structures. You put your toxin, and the cells will start to metabolize the toxin, but the toxic compounds will be secreted, and you will wash it every 2 days when you change the medium. When the cells are in 3D, those toxic compounds accumulate in between the cells and then start to kill the cells. And the best example we have is paracetamol, or acetaminophen in the US, which is a very potent hepatotoxic drug. If you can put paracetamol in hepatocytes in 2D, they will be fine. But if you put paracetamol hepatocyte going 3D, you will kill them for sure. So no, what I'm trying to say is that yeah, the 3D really allow you to model more complex mechanism that you have in the 3D.
Thank you. One final question is, the hepatocytes have heterogeneity. Even from the same person, they are not the same. How do you deal with that? Especially when you do IPS or mix other things.
Okay, so that, no. So heterogeneity, again, we need to define what is heterogeneity. So there is zonation. Okay, so zonation is like, when you take the architecture of the liver, the liver is divided in small lobules, and in those lobules, you have hepatocytes that are organized very precisely. And those hepatocytes will show a gradient of expression of key markers, which is based on oxygenation, glucose, and so on. Gradient between the central vein and the portal vein. And that's just the same hepatocytes, but receiving different information. And this zonation is important in the liver for functional, but because of the architecture. It's not really different type of hepatocyte or heterogeneous hepatocyte. It's just hepatocyte with different, again, patterning due to different microenvironments. So the question is more, can we model this zonation in vitro? And so far now, what we know is that yes, if we increase glucose, oxygenation, and so on, we'll see variation of some of those markers that are in zonation. Not yet perfect, but that's one of the functions we like to reproduce in vitro.
Thank you. Another question came in. What microenvironment component has the most significant impact on SC transcriptome profile of cholangiocyte organoids?
So really the key is the ballacid. That's really something that has a very, very strong effect on the cells in terms of transcriptome. Clearly, that's have a major impact on the cell, and cells really sense bile very, very efficiently. They're super resistant to bile. It's one of the rare cells that can resist to bile, but they can sense it very well. And for us, that's something that is really interesting, because that really changed the profile of the cells, and they react extremely quickly to that. That's the main factors we found so far. You can change those factors. You of course change a bit the profile, may block their proliferation and so on, but that's not what really make them a profile that will be more regional specific. It's really ballacid.
I see. Well, thank you so much for your time and for the great talk, Professor. I appreciate it. Have a nice
Thank you.